So this meeting is going to be recorded. If you don't want to be recorded or see your face, then please turn your video camera off. So listen, it's a great pleasure for me anyway to introduce Mats. We've known each other for a number of years. Mats, in his webpage, tells me that he started doing his PhD about heme biosynthesis and Bacillus subtilis before he moved into working with chlorophyll mutants in barley. I met him first when he worked in the Cambridge research labs in Copenhagen, where he was also working for a number of years on Barley Mutants before he moved to Lund University, where he's a professor and head of unit in molecular cell biology. Mats has worked on mutants of genes involved in a whole range of different things: plant architecture, surface wax biosynthesis, chlorophyll biosynthesis, amongst others. And I'm not sure, but for those of you who knew the late Uda Lundqvist, I always remember Mats as very often chaperoning Uda when she was in her 80s at various events, making sure that she was okay and able to participate fully in the meetings. And, you know, for me, that said a lot about the sort of person that Mats is. So it's a real pleasure, Mats, to have you here to talk to us today. I'm going to leave you to introduce the topic that you're going to talk about just in a second. Before I do that, I want to ask the audience, if you have any questions, please use the chat function, and we'll go back to any questions that you enter into the chat at the end of the presentation. So Mats, let's hear all about Barley Mutants. Over to you.
Thank you very much, Robbie, for the introduction and for this invitation. Now I should share the screen. And should go here. And I should press on that one, then something disappear for you. 12 o'clock in the image, there's something that is a little bit disturbing, but I think we have to live with that. And now I hope you see the first slide with
Perfect.
the title.
We see it perfectly. Yeah.
Great. So now you have my contact details if you want to contact me. And yes, I will talk about barley mutants and the gene hunting we are doing. And I guess all of you are working with barley, but if you ask someone who's not, this is barley just before harvest. It's a very common crop plant, diploid, which is quite convenient compared to wheat, for example. 7 pairs of chromosomes, has a big genome, reference genome published 2017, and we do have a lot of mutants. And here we have some of them. Picture A. Oh yeah, I should do this. I should get the red dot as well. Now you should see the red dot. So here we have chlorophyll mutants. So this is sort of wild type. And we have light green mutants and yellow mutants and white mutants. And I like 350 plus of chlorophyll mutants in this collection at Nordian, the Nordic Genetic Stock Center, where most mutants are coming, I'm working on. There are like 750 anthocyanin and proanthocyanin mutants, not making anthocyanin like this. There are a lot of morphological mutants, so there are more. There are like 5,000 mutants with compact spike, pyramidatum, dense spike mutants, and so on. There are broadleaf mutants, there are dwarf mutants compared to wild type, there are giant mutants compared to wild type. There are mutants where you get many tillers. If you plant one seed, if you plant one seed of these mutants, you just get one tiller coming up. There are also early maturity mutants. Here's some MAT-B7 mutants compared to bonus. Waxless mutants, you can see they don't have they're more pale green than the bonus. And yeah, these mutants are very useful, and I'm working a lot with them. The reason we have them is that Åke Gustafsson, 1928, together with his supervisor, Herman Nilsson Ehle, they started to induce mutations in barley with X-rays, and then later also used chemicals. And Uda Lundqvist, who Robbie mentioned, she was a disciple of Åke Gustafsson, a student of him, and she made sure that all these mutants ended up in the Nordgene collection. My favorite mutants, yeah, Robin mentioned some of them: chlorophyll mutants, chlorophyll biosynthesis and chloroplast development mutants. I like the plant character mutants, early flowering mutants. And today I think I will tell you about the chlorophyll mutants mostly. Some people don't believe that there are so much applied interest, but I think they could be, and they're at least very interesting to study from a scientific point of view, because I think you can do things with barley which you can't do with Arabidopsis, for example, just because we have a big seed, lot of starch, so they can promote the growth of lethal mutants. I showed you some of the chlorophyll mutants in the previous slide. So we have white mutants, and they're called albina. We have yellow xantha mutants. We have two kind of light green mutants, viridis, they are the mutants which are lethal, which, yeah, like the xantha and albina mutants, because without chlorophyll you can't survive. So they die normally after like 10 days when the starch has been consumed. But then we have the chlorina mutants, which are light green, and they are viable. And then we have also striped mutants, tigrina mutants and striata mutants. So working with the lethal mutants requires some sort of logistic and planning, because the homozygous mutants, you can't keep them in the seed stocks. So you always have to work with heterozygote seed stocks carrying the mutations, and you can't normally differentiate them from the homozygote wild type. So every time you do some planting, you have to sort your plants and the seed. And yeah, I will tell you a little bit more about that before. So this is what photos from such a sorting experiment. So we put a spike from a plant into vermiculite, and then we let them grow. And this was a segregating xantha mutant. You can see we have yellow, nice homozygote mutant seedlings here among the green. This is an albina mutant with a white, and there we have a tigrina mutant being striped. So I have actually this collection. Not so many of the chlorophyll mutants are, they're not actually available from the Nordgene collection. They do have the stocks there, but since they have problem to do the propagation of them because of these concerns, you have to have the heterozygote stocks. I have them here, and yeah, so I in principle have the stocks. And the reason I have them is that I was postdoc at the Carlsberg laboratory with Dieter von Wettstein, who was also a student of Åke Gustafsson, and by the way, a younger brother to Udda Lundqvist. And I was a postdoc there 95 to 97. And I was his last postdoc. So then he asked me, "Oh, do you want my collection of barley mutants?" And I said yes. And then he moved to Pullman in Washington State and worked there for another at least 10 years. My work has mostly gone on in this part of the world, quite close institutes, close to each other, but still international experience between Denmark and Sweden. So Copenhagen has the Carlsberg laboratory here. I'm here in Lund now. So after my postdoc, I went to the Department of Biochemistry, where I was for 11 years. And there I mostly worked on the magnesium chelatase mutants. So a very defined set of mutants. For the magnesium chelatase, it's a protein complex consisting of 3 gene products. So xantha H, xantha G, and xantha F. We have 7, 5, and 8 mutants there. Yeah, we did a lot of nice and interesting studies there, I think. Then after that, I moved back to Carlsberg laboratory and worked there for 6 years. And there I expanded my interest to other mutants than chlorophyll mutants. And now since 2015, I'm back at the Department of Biology. Yeah, well, back to Lund University, but in a different department. So biochemistry before, now biology. So there I'm sitting right now. But now to the gene hunting. So Already, yeah, when I started as a postdoc in Dieter's lab at Carlsberg, my task was to identify the genes deficient in mutants. So Tigrina D12, that was my postdoc project. And it was way too early actually to identify it there in 1995 to 97. And back that time, we very often use some kind of educated guess approach to identify genes. We try to, yeah, in Lund, when I moved back to Lund to biochemistry, we try microarray, develop microarray methods to identify the genes deficient in the mutants, but it never took off. We spent so much time on creating the libraries. Classical map-based cloning with F2 populations we have tried, but now we have landed in this kind of concept. We still do F2 mapping population, but then we do genome sequencing of pooled F2 plants. And we have a good pipeline here now. So from being the bottleneck in our work, this is not really our bottleneck anymore. Our new bottleneck is then to do something with identify genes to make it worth to publish them. And yeah, the first step we have to do is to make a mapping population. We take a mutant and we normally cross it to quench. Cultivar quench is Scottish malt quality, malt barley. And our mutants, they're often done in bonus or forma or something like that. And we know by experience that quench and bonus, they have very different genetics there. So we have a lot of, or at least enough of SMP markers. So we just get the F1 generation, having half of the mutant and the quench genome. And then we just do the selfing and we come to the F2 population and we have this shuffling around of gene segments that happens there. And most of the time we're working with recessive mutations. So in this plant, for example, we have, it's homozygous for the mutation. So this one should show the phenotype. This one should as well. So in principle, we grow the plants and look at the 25% showing the mutant phenotype. And yeah, so normally it looks like this when we do the planting. So this is where we were hunting for the early maturity gene EAM8.K, which we had a mapping population cross to Bowman. We planted like 1,000 plants here, one seed in each individual pot. 10 days later, we had 2 leaves. After 44 days, we started to see them flowering, so we could do the phenotyping. But then we work with chlorophyll mutants. It's not so straightforward when we do the F2 population, because I told you about the lethality of the mutants. So these plants, they die. So what we have to do, we have to do crosses with green plants. And we can't distinguish between the wild type homozygous or the heterozygous plants. So we just do a cross with something green, being the mutant and quench. And then we have to grow the F2 generation to get the F2, and there we have to do sorting. So there we typically sacrifice one spike per plant, put in vermiculite and see if that segregates. And if they do, then we know that the F1 mother plant was something we want to keep, and then we throw out all the others. So that's a drawback, which makes the work a little bit more heavier when we work with these chlorophyll mutants. But on the good side is that we definitely don't need to do that in the greenhouse when we grow the F2 mapping population, because instead of having a big greenhouse with individual pots, we can put all the 500 seeds in this little box, which is maybe 20 times 20 centimeter. And after 1 week, they grow like this, and you can see it's very easy to distinguish the yellow homozygous mutants from the green material. So yeah, we can plant like 500 F2 seeds in this little box. Then we collect the 25% of the yellow or white or striped leaves, depending on what kind of chlorophyll mutant we have. And then we pool the leaves together and isolate the genomic DNA. So this is DNA from a pool. Then we send the DNA from the pool to whole genome sequencing, and we get the data back, and we start to map the SNPs and look for linkage. And I should say that the pipeline has been done by our PhD student here, David Stewart. He will defend his thesis the 28th of September. And if you have some money, you should call him and try to get him, because he is really, really good. So this is from David. So here we have 1, 2, up to the 7 barley chromosomes. And what appear to be black here is actually dots. So you can see some regions we have a lot of black dots, so they are rich then. So the difference between quench and bonus in these two cases, we have a lot of SNPs here. Then there are some regions where we don't have so many dots, but it's definitely enough. We have a lot of dots representing different SNPs. So what you're looking at here is where we did mapping of the Albina C12 mutation and the Xantha I69 mutation. And David did then kind of an average of these dots here over a certain window. So we have a yellow line here. And when the line hits the roof here, then we know that we have 100% linkage. So in chromosome 1 here, we don't have anything, but then suddenly something happens here in the chromosome 2, because here we hit the roof all the way here. So that tells that the Albina C12 mutation is located in the centromere of chromosome 2. If we go down to Xantha I69, nothing happens here. That is very exciting. Until in the very, very end here, it goes up here. So this mutation is in the telomeric end of chromosome 7. So then we have done the mapping, but because we are sequencing the genomic DNA of these mutant pools, the mapping population, it's very likely actually that we have the DNA sequence of the mutation. So what David then did was that this is a message table, but I will talk you through it. He looked for all homozygote mutations in genes on chromosome 2. In the case of Albina, I go back one slide. So in the case of Albina C12, he looked for all homozygote mutants in the entire chromosome 2. And we found 15, so I think this should be 11 of them, but it's enough to show you the principle. So here you can see all of them are located on chromosome 2. The position of this mutation, which is an A instead of a G in the reference, is at position 7,924,412. And it's a high confidence gene, kinase family protein, but this one was not in the interval. And then we have a few more which were not in the interval. So here we have 1, 2, 3, 4, 5. 5 which were in the interval. And this one, polyprotein something, but this seems to be some sort of repetitive DNA, so we ignore that. This one, transmembrane protein 53, low confidence gene. Next one is also low confidence gene, high confidence gene, low confidence gene. So among these 5, it's only one being a high confidence gene. It makes it also very interesting that it has a piece of DNA that should encode a transfer peptide specific to thylakoid lumen. That's of course very promising if you were looking in an Albina mutant. So our candidate gene is then that the Albina C gene should be a pentatricopeptide repeat containing protein. And this one is then located in the centromeric region. In the case of the Xantha I, actually, I don't know if you looked at the psi chromosome 7 or just in the end, but anyway, these were not in the mapped region, so we only had one gene with mutation, homozygote mutation, in this region here, and it also had a thylakoid lumen transfer peptide coding sequence. and this is FTSH, so that's also related to chloroplast metabolism, so it's a protease. So from these two scans, then we have two candidate genes. So then we have to go to the next step. So how do we verify that they are actually the correct genes? And then we are lucky that Udda and Dieter and all these guys, it was very important that all allelic mutants are kept. So we don't have so many allelic mutants, but with albina C, at least we have 4, which is better than 3 and 2 and 1. Albina I, we have 3. So then we simply grow the allelic mutants and sequence them for the candidate genes we've identified. And yeah, because random mutations obtained by physical or chemical treatments, in these lines, this treatment causes a lot of random mutations all through the genome, represented by this line here. So line number 1 has a lot of mutations which are different from line 2 and line 3 and so on. But since they are allelic, so before they could do very much with these mutants, they could at least do crosses between them and group them in allelic groups. So if they're said to be allelic, there should be one gene where they should have mutations. That should be the common denominator to them. It should not be the same mutation, but different, but still in the same gene. So that is what we are trying then. Yeah, I said we had just 3 and 4 of these albina C and xantha I, but in this case, when we identified Eserifrim waxless mutants in the C, Q, and U gene, we had a lot of mutations, and you see all of these mutations are located in the 2 exons of the SERQ gene. Here we have just 1 exon in the SERC up here, SERQ here. They're all nicely mapped to the gene. SERU, we have 5 exons, and we have a lot of mutations spread all over. Yeah, so my message, if some of you are working at gene banks, please keep your mutant groups with many alleles. For me, they are the most important ones to keep because they help us in our identification of and verification of the candidate genes we have. Yeah, and yes, we were right. These were the genes in these mutants. Yes. Okay. So my view of a mutant research. So before, the gene hunting has been the bottleneck, and but since I have also some sort of biochemistry in my background, I also want to widen my research and see the effect of the mutation, to learn about the protein and the processes. So still keeping you on the thread here with chlorophyll biosynthesis. So here you have a schematic view of chlorophyll biosynthesis. So in plants, you start with glutamate as a precursor, which you attach to the tRNA, and then you get 5-aminolevulinic acid, which is also our cells also making this for heme biosynthesis, but we start with glycine and succinyl-CoA in the condensation reaction. But then we have a common branch up here, up to protoporphyrin. Heme is made by ferrochelatase. The chlorophyll branch, we have this magnesium chelatase I mentioned before, where magnesium is inserted. And then we come eventually to cyclase enzyme here, and chlorophyll A is the typical chlorophyll molecule. So I will give you an example of what we have done here with something related to the cyclase. So now you know where that is located in the pathway. And this is what it looks like. Here you have the substrate of the cyclase, magnesium protoporphyrin 9 monomethyl ester, and the enzyme adds this ring, so the 5th ring of the chlorophyll molecule. Then we get protochlorophyllide. That is done by the cyclase. Some history about barley cyclase. Before 1970, only mutants were known, so everything about cyclase was known from mutants. So it was found that xantha L mutants and viridis K mutants accumulate magnesium protoporphyrin monomethyl ester. So then you could expect that magnesium protoporphyrin monomethyl ester is the substrate of an enzyme where the Xantha L and Viridis K gene products are involved. 2005, we succeeded to identify the Xantha L gene. So this is the core product for the cyclase enzyme. And we succeeded to identify it at that time because advances that were made in bacteria. So Rubrivax gelantinus, they identified the ACSF gene and found that was a cyclase. So by doing degenerated PCR primers for this gene, we could amplify the Xantha L gene in barley and sequence that from the mutants. We have 3 here, Xantha L35, 81, 82, and confirm that they had mutations, changing amino acid residues here. The last one is a nonsense mutation introducing a stop codon. So that was the step we were for many years. But obviously, since we have the Viridis K here and we also did some biochemistry, it was obvious that we have other components involved. One question from the reaction mechanism is where electrons are coming from. And yeah, and of course, what gene is Viridis K? So again, David Stewart, so this is what he has done. He identified the Viridis K. He found a candidate gene in an F2 population that it was linked to the telomeric region of chromosome 4H in barley. And yeah, this is the structure of the Viridis K gene and the mutations. We have 7 exons. We have only 2 mutants in this. The Viridis K 170 is a G to A mutation in exon 4, changing amino acid residue. This one seems to be something big. Try to illustrate that here in B. So this is some PCR reaction mapping. So a filled line like this means that this PCR fragment we can make, and we can do some walking here with PCR primers up to a point here. So this we can amplify, but when we try a reverse primer here, we don't get anything. And from the other side, we have something similar, meaning that we have a region here where probably one of the inversion points is located, but we don't have the exact, we don't know it exactly at the DNA level. Yes, but the gene product, so it looks like the Viridis K gene product is encoding something called FDC2 in Arabidopsis. So this is a ferredoxin, and ferredoxins, you know from your biochemistry studies, something from the photosynthesis here. We have photosynthesis complex 2 here, complex 1, the cytochrome B6F complex, all the plastokinone and so on. We have the flow of electrons going up and down here. We have the pumping of the protons from the stroma side to the lumen side in the thylakoid. And up here you have the ferredoxin. So ferredoxin receives electrons from photosystem 1. Okay, please remember to type a question. Okay. And it transfers then its electrons to ferredoxin NADPH, NADP reductase that can catalyze the formation of NADPH from NADP+. So Viridis K is a ferredoxin, but there are many ferredoxins. So I mean, the one I showed you from the photosynthesis, that's the classical one, the so-called leaf-type ferredoxin you read about in the literature sometimes. So when we searched the barley genome, or David did, he found 8 ferredoxins. And one was then the FDC2, the Viridis K, and it's called C2 because it has a C-terminal extension compared to other ferredoxins. There is also an FDC1 in barley, and then there are other FDs here. So we think then simply that ferredoxin C2 is the electron donor to the cyclase reaction. So here we can look at the intermediates. So we think that the the reaction, the enzymatic mechanism here is something similar to beta oxidation of fatty acids. That we introduce a hydroxy group here on this carbon here, which is then further oxidized to keto group, and then we have a ring closure. So we have this. And in all these steps, we have the ferredoxin introducing electrons into the system. So the study of these mutants, the Viridis K mutants, has been very useful because they are named Viridis, so they are the light green lethal mutants. They can make a tiny amount of chlorophyll, but you can't really see it. Sometimes when you grow them in certain ways, you can. So trace amounts can be detected. But the interesting thing is that the Viridis K mutation can be complemented by the Barley FDC2, but not the leaf type barley, which we have tried with agrobacterium agroinfiltration. So you know this system which you normally see in species with more other leaf structures like Nicotiniana. So simply you make a construct in agrobacterium and you introduce the bacteria, you make a little scratch in the leaf and have a syringe and introduce that in the space between the cells in the leaf. So that we did here actually with the Barley Viridis K23 mutant. And you can see that, yeah, we do have some green here when we have introduced the Viridis K gene, the FDC2 gene. But when we do the same thing with the FD1 ferredoxin, we don't see any green. So this tells us then that FDC2 is actually the electron donors to the cyclase reaction, and it can't be done in vivo by other 7 ferredoxins you have in the barley genome. And this is purely results from the genetic studies here of this plant, because if you do the in vivo studies, which we have done as well, we can produce the xantha L protein and do reactions in test tubes, and there we can have any kind of ferredoxin, and the assay works nicely anyways. We can take any of the 8 ferredoxins from barley. We can even buy ferredoxin from Sigma, which is made from spinach, and have the reaction going. But there is something in vivo in the system that makes FDC2 essential. So I think this is a very nice example, how the chlorophyll mutants in barley can be used. And we can tell something which we can't really do with pure biochemistries only. Yes. Yeah, future cyclex experiments. How connection could the electrons coming directly from the photosynthesis? We don't know. Maybe we don't have a good system to set that up either, unfortunately. Yeah, when we do the assays, we think that the electrons are coming from NADPH, so then they should go via an FNR, ferredoxin NAP reductase, but we run the reaction reverse. And this one donates the electrons to the ferredoxin C2. There are some special ferredoxin reductases, FNRL, so we would like to knock out that with CRISPR, for example. And of course, we would also like to do structural studies with the xantha L Viridis K. I haven't talked about YC54, which is another component, probably a chaperone for the folding of the xantha L, to determine the protein structure with X-ray crystallography or something. So that is things we would like to do with the cyclase in the future. And then I will check how I'm doing on time here. Okay, I do this part as well. Can the old mutants be used for anything good? Yeah, maybe many of you have an applied view of barley research. And these old mutants, yeah, I think there are some good things here. I think the mutant at K32, causing a short, calm, sturdy, lodging resistant, is a good candidate that could be used in plant breeding. I also believe that the early maturity mutants could be good. Do we actually need the orns in barley? So maybe the LKS1A could be used. Elongated outer gloom mutants, you get some gloom, some more leafy structures around the kernels. Maybe they can increase the starch production in the very end of the development. And they are found in the EOG1K mutations, for example. But we have the problem with sexual reproduction. Why should you have sex and risk that your perfect gene variants are not transferred to your offspring? Which is what this is about, because if we have today's best cultivar and we do a cross with this old mutant, having a desired trait, we transfer all of the maybe not so good DNA to the offspring, and the plant breeder need to do a lot of back crosses to get rid of that. Oh, there are questions coming here. I'm supposed to answer them directly, or we take them after?
At the end.
Yeah, okay. Yeah, so what the breeders would like to do is some kind of gene stacking, or rather allele stacking. So they want to have the elite cultivar, add that specific mutation. When that is done, they want to add another one and another one and another one. And of course, these kind of tools are approaching here with CRISPR, so it should be possible to do it soon. And I mean, CRISPR we haven't had for so many years. Is it 11 or 12 years now since it was invented, discovered? So where are we 20 years from now? Then maybe this is with a snap of your finger, you can do this very easily. And of course, that would make all these barley mutants not so interesting anymore, because then you can knock them out and find out gene functions anyway. But still we'll need to learn which genes do we need to stack, which alleles do we want to combine. And I would say that very much today we are at the one gene level, with maybe exceptions of some resistance genes. So we know that two genes in one pathway, how we should regulate them in relation to each other. But otherwise, we know that, okay, we can knock out that gene in early maturity, we can knock out that to affect spike density, whatever. But to have two genes or three genes in the same pathway, we are totally lost. So we definitely need to learn more. And I think these old mutants, they will not be old and unfashioned until we have reached at least a 2-gene level or a higher level. And also lethal chlorophyll mutants can guide us to genes to modify in some way. So my conclusion about the mutants in the Nordian collection and other collections is that they are actually a fantastic resource for gene identification. And we can learn a lot about useful alleles, although I don't think really that they will be so important for the actual breeding, because breeders are hesitant to get not so good alleles in other genes into the elite cultivars. And yeah, I have to acknowledge a lot of people. David, as I said, has been very essential, and Shakira, as always. Heme has been involved in this as well, and others, and we got some money to do the work, so thanks to them as well. And thank you for inviting me, and thanks for listening. Oh, applause here. Mm-hmm.
We're all trying to give applause by using some kind of digital
Yeah.
thing. Matt's fabulous. Well done.
Okay,
I
thanks.
think the floor is open for questions. There's at least a couple in the chat. Let me just. Shall I read them out, Matt, and then, or I'll just ask the people that wrote them to
Yes, please do
that.
So
I'm going to read out the first one that I wrote, which I remember some time ago we tried to write a grant on chlorophyll deficient mutants,
Mm-hmm.
and we had a slightly different strategy from what you described. But we were kind of naive at the time because we didn't know how many independent genes might be involved in the process. So all that you've done now, do you have an estimate for the number of independent genes that would generate a chlorophyll deficient mutant phenotype in barley?
You mean how many genes there could be to generate a chlorophyll deficient phenotype? Yeah, well,
I'm trying to understand the number of genes that are actually involved in the overall pathway.
Yeah, to start with, the 357 mutants I got from Dieter von Wettstein, they represent 105 loci.
So allelism tests have been done.
Yeah, so at least 105, but probably more. I guess there should be at least the double.
Okay.
Yeah, and then if you count all, it should be many, many ways to get pale green mutants.
Okay. So there's still a lot to do.
Mm-hmm. Yeah.
So the last part of my question was that do the barley mutants give us a unique insight into photosynthesis in temperate cereals, as opposed to, you know, whatever's going on in rice or in Arabidopsis or whatever?
I think the mechanism will be the same, regardless what kind of plant you're studying, actually. But I think the unique thing with, because if you generate chlorophyll deficient mutants in Arabidopsis, you know, you have the tiny small seeds that can't really support a big plant, so you have limited access to do biochemistry with the material. You can grow Arabidopsis seeds on sugar media, right? But very often, most of the chlorophyll mutants I know of in Arabidopsis, they are leaky. I mean, they're light green.
Okay.
So we have these clearly yellow plants, which are not so common in Arabidopsis. So, I mean, there we have an advantage.
That's what we want to hear in this meeting.
Yeah, I think so, yeah.
David, do you want to ask your question?
Yeah, sure. I'm here. Yeah, well, in one of your very nice talk, by the way, I really appreciated it. Thank you.
Oh, thank you.
On one of your early slides, the one where you were showing the peaks of the differences between the allele frequencies in the pools. I noticed that on both of those crosses, both of those mutants, there was some kind of curious, let's say, background spikes on chromosome 5H.
Yeah, I don't know what's going on there.
The question is that because I mean,
yeah, I will show it. Slide 50. No, that was later. It was this one. Yeah, chromosome 5 here. It goes up here. No, I don't know why it is like this.
Is it? I mean, these are both the same crosses between the same backgrounds, right?
Yes, they're both from Bonus. And actually, David is comparing, he's using this one as a control to this one and this one as a control for this one because they're both made in Bonus. So we
can't see the slide, by the way, Mats.
Pardon?
You've got the slide on your own screen, but we can't see.
Sorry. Yeah, sure. Yeah, I shouldn't be so selfish. I should share my screen. Yep.
Yeah, I mean, in this case,
so here, right?
Yeah, exactly. And you know, I'm not sure that it's exactly the same shape that I see, but we see pretty consistently for various crosses, but not that I've seen anything like as many mutants and as many pools as you probably have. But yeah, we see some peculiar stuff on parts of chromosome 5 in quite a lot of crosses, and sometimes we think the mutants are actually there. And I was just wondering if that was a consistent pattern that you've seen even with other crosses, or
I think we have actually. So we've been thinking if this is something specific that happens when we do this to quench, because we have only used quench as cultivar in the crosses, because we know it works so well. Yeah. No, I can't tell you, but I mean, it should not be something real here. It should, because these are monogenic.
No, for sure. In this case, the hit is on is on 2 and there's not much recombination, so the interval's enormous. But it's that thing on chromosome 5 that makes me curious. I mean, there is also some bands of very marker poor bands in the distal end. No, I was just curious.
Yeah. No, we don't know what's going on here, but we have noted that, yeah, it does a jump up here.
I should say, if anybody wants to chip in with an explanation, then please do. I mean, it's obviously segregation distortion there.
Yeah.
Do you know which parent the more frequent allele comes from? Is it from the mutant or from the wild type? From quench or from bonus?
I don't know. No, I don't know.
Okay. I think we'll have the opportunity to discuss with Mats at some point in the future.
Yeah, I will ask David if he has some idea.
Cool.
David Stewart, my student.
Fantastic. Cheers.
Okay, thank you. Yuncheng, do you want to ask your question? Or would you want me to do it?
Can you hear me?
I hear you, yes.
Oh, yeah. I just wonder, since you mentioned that the FD2 or FDC2 has an extra C-terminus domain, and have you made some truncated version of the protein or mutagenesis to determine what's the function of the C-terminus? Is it necessary for the in vivo or in vitro functions?
No, we haven't, but my guess is that it would not have an effect on the in vitro function. No, but we haven't done it. But my guess is that it would perfectly work in our in vitro assays, even without the C-terminal tail. So it must be something specific, probably interaction with the Xantha L, for example, or something else. You can always say regulation.
Do you think it can determine the subcellular localization, or?
The subcellular localization, yeah, maybe. Yeah. So far it doesn't, when we try to produce it, it has a tendency to form inclusion bodies, but it's not really a membrane bound. To some extent it is, it's a little bit sticky. That's not been so nice to us so far when we try to do the biochemistry with it and produce it as a recombinant protein.
Okay, thank you.
Thanks.
Okay, Maria.
Hi, Mats. Thanks for a nice talk.
Oh, hey, thanks.
I probably have a similar question. I was wondering if you have any clue why this FTC2 is different in vivo from the other FDs. I mean, based on sequence, based on whatever. Any ideas?
No, we need to find that out.
Okay, good.
Yeah,
thanks.
Yeah, exactly. Yeah.
Sounds good. Yeah, something to Thanks.
So, thanks, Maria. So that's the end of the questions in the chat. Does anybody else in the audience want to stick their hand up or just shout out with a question? I can't see everybody, unfortunately, so. No? Looks like you're getting off the hook, Mats. I can tell you some information about the chromosome 5 area, maybe offline if you like, which might help. I don't know if it'll help at all, but I can tell you some information that we have on the chromosome 5 centromeric region. Just for everybody that's on the call, though, I just want to mention that the next seminar is in the chat is on the 8th of June, and it's about a transcriptomic atlas and genome dosage dependent development of barley grains, and it's by Alice Pichinka. So you can sign up for it on Eventbrite. I'm hoping that everybody is going to get an invitation on the morning of the talk anyway, where you don't have to sign up, but we've still got to work out how to do that. So Mats.
Yeah, I want to say one thing I forgot. I should say, now when I have a barley audience here, I have a postdoc grant if someone needs to do something in Lund for at least 1 or 2 years, you're most welcome to call me.
Is that open to all of us?
You're most welcome, Robbie. Even I would consider you.
That'd be great. Listen, to everybody, thank you very much for turning up and enjoying Matt's seminar. And thanks again, Mats, for a great talk. You look after yourself, and everybody take care.
Yeah, thanks. Bye.
Bye-bye.
Bye, everybody. Ciao.
Ciao.